Right, today's presentation is on nutritional consideration for citrus, and that is to try and achieve high yields with exceptional quality. So firstly, we need to understand what is our goal in citrus production, and then we will cover aspects such as efficient mineral plant nutrition concepts, such as pH, such as how much fertilizer do we apply, what stage of the plant do we apply the fertilizer, and what sources of fertilizer do we need to apply. And then at the end, we'll just touch on the possible benefits of potassium as a foliar application. Firstly, what we're trying to achieve are optimum yields, high yields, and with the best fruit quality. Now from this slide, you can see what the various quality and yield parameters are, so I'll just run through them briefly. We have fruit size, fruit weight, fruit set, number of fruit per tree, and that all affects your yield. And then you have certain quality parameters such as peel thickness, peel blemishes, sugar content, which is TSS, the fruit acidity, the acidity to sugar to acidity ratio, and the ascorbic acid content and the juice content. You just need to be aware that in your area there might be slightly different sugar to acid ratios or different total sugar contents. For example, if we compare Brazil and in Florida, the total sugars is 12 for both areas, but in Florida they prefer a sugar to acid ratio of 14 to 16, and in Brazil 12. So it's obvious in Florida that they prefer their oranges sweeter. If we go on to the first of the plant nutrition concepts, and if we look at pH first, we always start with a pH. One needs to take a soil sample and see what the pH is of the soil. And we have a pH range of 0 to 14, 7 being neutral. But you need to remember that pH scale is a logarithmic scale. So 6 is 10 times more acidic than 7, and 5 is 100 times more acidic than 7. So just remember that the pH scale is a logarithmic scale. This picture on the left you've probably all seen before out of textbooks, but it shows the nutrient availability at different pH ranges. So if we look at the elements individually, well, you can study this in your own time, but you'll see if the acidity is too much, then nutrients become less available. If the acidity is higher or too high, then nutrients also become unavailable. So what is the ideal pH? It depends on the method of testing. If you use a KCl scale or testing method, the ideal is about 4.5 to 6 pH units. If you use the calcium chloride testing scale, then the ideal is 5 to 6.5 pH units. And if you have a water testing scale, then your optimum pH is between 5.5 and 7. A good example of an element which becomes unavailable at a pH is iron. That's normally the first to become unavailable, and if you're testing with a water pH, you will see that at a pH of 7, iron becomes unavailable, and your plants will show yellowing and deficiency symptoms. The graph on the right, on the x-axis, is the pH scale measured in calcium chloride, and the tree in tons per hectare is shown on the y-axis, and this was for oranges in Brazil, and you can see that as the pH increased from 4.5 to 6.5, so the optimum pH was reached and the yield increased on a per hectare basis. The next concept is dosage, or how much do I apply, how much fertilizer do I apply. You've probably all seen this picture shown here, which shows all the nutrients in a barrel form, and if there is one nutrient that is missing, it becomes the weakest link. So you can have all your elements in optimum amounts, but if you have a deficiency of one, you are going to affect your yield. So what dosage do we need to apply? Firstly, we need to look at the plant requirements. So here's a list or a table of various citrus types, and then on the table also shows the nitrogen, phosphate, potassium, magnesium, calcium and sulfur that is removed per metric ton of yield. And this is the starting point for all recommendations. You need to understand how much nutrition or how much mineral elements are actually being removed by the crop, because that's what you need to apply. You also need to consider inefficiencies. You also need to consider nutrients possibly going into root growth, or possibly also being lost through prunings. This table that I show you now is leaf analysis, also a very, very important tool to establish what your fertilizer recommendation should be. And leaf analysis should be taken at the same time every year, and it should be taken from a particular height, like shoulder height, and it needs to be taken from the same kind of shoots every year, so that you get a good idea of what's happening in your orchard, and also a representative sample. So you take many trees to make up a subsample from which a sample is taken, and that gets analyzed. The result you get back, you need to compare with the norms, because there are norms. You need to just be aware that in certain areas the norms might differ slightly, and for certain citrus types the norms might differ slightly. But it's a very useful tool, and it's something that you need to use. If we touch on the elements individually, we start off with nitrogen. Nitrogen is the second most utilized element, and it's taken up as nitrate and ammonium, but we'll show you later preferably as nitrate. Accurate nitrogen application is the most important in all aspects of growth and fruiting. The amount of nitrogen should be supplied according to citrus type, variety, age, and the target yield. Nitrogen is the main component of amino acids and proteins. And a lack of nitrogen, we all know, leads to very poor tree vigor. And the excess of nitrogen can have an effect of quality parameters. And I've shown you one graph here, the sugar to acid ratio, where you can see the nitrogen rate on the x-axis, and you can see the sugar and the sugar to acid ratio on the y-axis, and the dark bars is the acidity, and the light bars are the sugar content, and the line is the sugar to acid ratio. And it's important to see that the sugar stays the same if you increase the nitrogen, but the acidity increases dramatically if you increase the nitrogen too much, and it negatively affects your sugar to acid ratio. If we look at phosphate, phosphate is needed in very small amounts, or relatively small amounts compared to the other elements, and it's taken up as the phosphate anion, H2PO4 minus or HPO4 2 minus. But phosphate is very vital in the plant's metabolic functions and energy functions. The uptake peak is during the early stages of root growth, going into flowering, and then the uptake of phosphate declines slowly during fruit development and to fruit maturity. So it's very important to have that phosphate on early. Also, it can affect quality, because an excess of phosphate can decrease peel thickness, but it has a great effect on yield and juice content. And the two graphs I show you now are the phosphate content on the x-axis applied as kilograms per hectare, and on the y-axis we have the fruit yield in tons per hectare and the juice content in percent. And you can see that as the phosphate application increased, so the yield increased of the tree, and also the juice increased of the oranges. The next element is potassium, and this is by far the most important element, because it is the most utilized element, and it's taken up as potassium cation, or K+. Potassium is used for osmoregulation, and that's transpiration controls the stomatas of the plant. It also is involved in photosynthate transport, nitrogen transport, and many other vital functions. Potassium has no structural function in the plant, only metabolic functions. Potassium has a direct effect on fruit retention, influencing yield. So that's your fruit set, and it also affects fruit size and fruit quality. If you look at the graph on the left, the fruit number per tree, that's your fruit set, and we have a control, which is the light bar, and a potassium nitrate foliar application is the middle bar, and potassium sulfate to the swell is the right-hand dark bar. And you can see with the potassium applications, be it foliar potassium nitrate or soil applied potassium sulfate, that the fruit number per tree increased. The application of potassium also decreases creasing and plugging, so it has a quality effect. And if we look at that percentage on the left hand or the y-axis, and we look at the increase in potassium in kilograms per hectare on the x-axis, you can see that the creasing decreased dramatically and the plugging also decreased with an increase of potassium. And the graph on the right, we can see the potassium effects on the ascorbic acid content, and these were tested in orange and in mandarin. And you can see that as potassium increased, the rate increased, so the ascorbic acid increased in both oranges and in mandarins. Before we move to the next element, we need to just touch on an aspect which is the N nitrogen to potassium ratio. And it's very important to understand what the correct ratio is of nitrogen to potassium, and it depends and it varies between phenological stages. So the balance definitely affects vegetative growth. It affects the development of reproductive organs, that's the flowers. The balance or the ratio also affects the filling of fruiting and reserve organs, and also influences the translocation of photosynthesates, and also has an effect on the maturation of the fruit and the post-harvest fruit quality. So if we look at these three phenological stages, we've got the vegetative stage, we've got a sort of a neutral stage, which is your flowering stage, and then we've got a fruit development and maturation, we call the generative stage. And the vegetative stage, the N to K2O ratio should be 1:1. In the neutral stage, the K2O should increase, so it's N of 1 and K2O of 1.5. And in the fruit development, flowering and or what we call the generative stage, it should be 1 nitrogen to 2 to 3 K2O, perhaps even higher. If we look at some of the secondary macroelements, the first one we'll start off with is calcium. Calcium is the third most utilized element, and it's taken up as the calcium cation, that's Ca++. And calcium is taken up primarily in the root tips of new root growth. Calcium is an important component and structural component of cell walls, increasing peel strength, increasing fruit life, and also leaf resistance to diseases. So it leads to a reduction of splitting and creasing, and also your rots. And the best movement of calcium into the fruits is during early fruit enlargement, that's after fruit set, until about color break. I have two graphs here. The graph on the left is calcium and peel strength. So this is tested with a penetrometer to test the strength of the skin. And you can see that with applied calcium, that's the dark line, versus the control, which is the light line, you can see that the peel strength increased with the application of calcium. The graph on the right just shows when calcium moves in the plant. So you have on the y-axis the calcium flux, and on the x-axis you have the days from fruit set. So you can clearly see both in mature trees and in young trees that the movement is from cell enlargement until color break in citrus. The next secondary macroelement is magnesium. Magnesium is required for various physiological processes, and it is strongly linked with the chlorophyll and protein production, that's photosynthesis. Deficiencies and excess will impact on yield and quality parameters such as TSS and acidity. I've got two graphs here. The first graph shows an increase in magnesium on the x-axis, that's in grams per tree, and on the y-axis we have fruit yield kilograms per tree. And you can see where the magnesium rate was increased, then the yield increased in grams per tree. And they also show you the single fruit weight that will also increase with the increase of magnesium applications. The graph on the right shows the sugar content and the acidity of oranges. This trial was done in Egypt, and the orange bars are the sugar content, and the dark red bars is the acidity of the fruit, and then the line is the TSS, or the sugar to acid ratio. And again, you can see with magnesium that the sugar content stays pretty much the same, but with too much magnesium you increase the acidity too much and you get a detrimental decrease in the sugar to acid ratio. So you need magnesium, but not too much. The next secondary macroelement we will just touch on is sulfur. Sulfur is part of proteins and enzymes, and it's essential for plant growth and development. It's required in very small amounts, and the removal by fruit is small. The one graph I've shown you is that a sulfur deficiency can lead to fruit drop. So if you look at the graph, you'll see a fruit drop in percentage on the y-axis, and you will see a control where there's no sulfur, and you will see two applications or two different products, ammonium sulfate and calcium sulfate, which both almost halve the fruit drop. So this leads to greater fruit set and greater yield at the end of the day. If we touch on the micronutrients, we have identified 6 micronutrients that are essential. There are other micronutrients that are beneficial, but perhaps not essential. So the essential ones are boron, iron, manganese, zinc, copper, and molybdenum. Micronutrients are required in very small quantities. They have a metabolic function, and some have a structural function, and some have a metabolic and structural function. But because they are required in such small amounts, it's perhaps better just to highlight some of the deficiency symptoms. And that will be decrease of fruit set and fruit weight, decrease in sugar content, decrease in ascorbic acid content, that's a quality parameter, decreased juice content, increased plant yellowing, as I said with the iron deficiency, that your plants will go yellow very quickly, and a decrease in photosynthesis and effective leaf area, and a decrease in tree vigor. So that's some of the symptoms that you will find with micronutrient deficiencies. It is very important to know your area, because certain areas are well known to have a zinc deficiency or a boron deficiency. So get to know your area and ask your agronomist, what are the common micronutrient deficiencies in this area, and rectify them before you have problems. I'd just like to touch on irrigation. I mentioned it now in the last pictures, but it's a very important part of plant nutrition. And it's very important to understand where your roots are. What's your rooting depth? And what is your water holding capacity of your soil? Now you can see your rooting depth by digging holes around your tree. You can see your roots are going to 60 centimeters or 80 centimeters. So you can establish what is my effective rooting depth. You can also take a soil analysis and send it to the lab, because they can calculate what is your water holding capacity of your soil. A clay soil will obviously hold a lot more water than a sandy soil. So you will then know what your rooting depth is, what your water holding capacity is. You will know what your field capacity is, and you will know approximately what your permanent wilting capacity is. That's when your trees will start to wilt. Most growers will irrigate to sort of 50% depletion between field capacity and permanent wilting point. The points that are important, as I've said, is firstly your soil rooting depth, but also your soil osmotic potential. In other words, if you've got a saline soil, if you've got a certain amount of salt in the soil, you perhaps need to modify or reduce the depletion, because your salt in the soil will be competing for the nutrients and for the moisture with your tree roots. So you want to maybe deplete that to your depletion will only go to 25 to 30% instead of 50%. Also, your management style and intensity. Some growers are really top notch, and they can manage a drip irrigation system where they've only got 1 or 2 or 3 bulbs per tree. But that's very intensive management. Some growers prefer to have full strip irrigation, so they can manage that more easily. So understand what management style and what sort of intensity you want to go to with irrigation. And then also, it's very important to use the tools that are available out there, such as neutron probes, such as tensiometers, such as evaporation pans, lysimeters, but use them. Those tools are there, and they're an essential part of irrigation, and irrigation is an essential part of plant nutrition. The graph I have here is just to show yield improvements with fertigation versus broadcast applications of fertilizer. And the 2 areas were oranges in Israel and grapefruit in Florida. And you can see in both cases the yield in kilograms per tree increased with fertigation as opposed to broadcast fertilizer applications. So if you can do your fertigation, that is beneficial, but then your irrigation also needs to be top notch. When do we apply the fertilizer? What's the right stage to apply fertilizer? So we've looked at the pH, we've looked at maybe the rate, and now we'll come to what stage do we apply the fertilizer. This picture shows 6 growth stages of a citrus tree. So you've got the pre-bloom stage, the full bloom stage, the post-bloom or fruit set stage, and you've got the fruit growth stage, and then you've got a later fruit growth stage until color break, and then you've got the fruit coloring going into the maturation stage. So those are probably the 6 most important phenological stages of citrus. But from winter to spring to summer to autumn, we're going through a floral induction and initiation. Then we go for a shoot growth, a spring growth flush. Then we have a root growth flush. And then we have flowering and fruit set and a cell division stage. And then we go into the summer growth flush and then a cell expansion. And then we go to a second root flush stage. And then an autumn growth flush. And then we go to the maturation. So that's how it works. You're alternating between different processes in the plant during different times of the year. This graph shows the development of a fruit, citrus fruit, in weight, and it's from fruit set until maturation. And you can see that it starts off slowly, then it accelerates, and then it tapers off towards the end. So that's a typical sigmoidal growth curve. If you look at the accumulation of the macronutrients over time, you can see that it follows pretty much the same curve. But as I said earlier, it's important to notice how much potassium is required by the plant relative to the other elements. So the potassium is much higher, then it's nitrogen, and then it's calcium, and then much lower down are magnesium, phosphate, and sulfur. But you can see how it accumulates also in a sigmoidal type of growth pattern. The trace elements as well, also an accumulation over time. Iron is the most important, then boron, and so you can study that graph yourself. This is a graph which shows the uptake of the nutrients by month. So it's actually more important than the total accumulation we've just seen, because this shows us when you need to put your fertilizer onto the tree. If you take a good look at the graph, and the purple lines are the nitrogen, and your peak is sort of November, December, January, February, then tapering off. This is also the southern hemisphere. If you look at the phosphate, the peak is earlier, sort of November, December, January, tapering off. If you look at calcium, that's the blue graph, that's also your peak is earlier, around December. And if you look at potassium, your peak is around January. So, but what is apparent from this is that all your nutrients are taken up primarily in the first half of the physiological stages. Your fertilizer also needs to get to the root zone, so one needs to put the fertilizer on even before this period. And from graphs like this, you then determine your recommendations and your split of nutrients. This graph, this table shows nitrogen, phosphorus, potassium, and calcium. And the difference, they've given 3 stages here: pre-bloom to flowering, cell division, and cell expansion. But you can see that all the fertilizer is predominantly put on the pre-bloom to flowering stage. And that's because of the uptake curves of the different nutrients. Potassium is probably the one with the longest spread going into the cell expansion stages. Now we come to a plant nutrition concept, and we will discuss the source of fertilizer to use. This is one of the most important aspects of this presentation. And to do that, we need to just study hydroponics for a little bit. We use this model as a model because, you know, your citrus trees, they need a nutrient solution around their roots. Your plants don't eat fertilizer, they drink fertilizer out of the solution. So we can look at a hydroponic model to understand the correct balances that is in hydroponics, and that should also be around your roots. The model I propose here is from Steiner, and the units are in millimoles charge per liter or milliequivalents per liter, but the units aren't important here, it's the concept of balanced nutrition. If you look at the 2 triangles, you'll see the triangle on the left shows the cations, potassium, calcium, and magnesium. And according to Steiner, for an ideal nutrient solution, there needs to be a certain balance between those cations. If you look on the graph on the right, you have your anions, you have the nitrate, the phosphate, and the sulfate. And also for an ideal nutrient solution, you need to have a balance between your anions, or a ratio, correct ratio. I think Steiner proposed this model because he could take a bean seedling and he could put it into this ideal nutrient solution, and it would grow, it would flower, it would produce fruit, and it was a perfect nutrient solution. You might have heard of other ones, such as the Hoagland's solution. If you've studied a postgraduate or done nutritional courses, you also get a Hoagland's solution, which is also a hydroponic solution, but the concept is exactly the same. So those balances of cations and the balances of anions are very important to have optimum growth and fruiting. But we all know that there can be a slight variation depending on phenological stage, as I said earlier with the nitrogen to potassium ratio, for example. So those ratios can vary depending on phenological stage, but the concept stays the same. You need to have a balance of the cations and a balance of the anions. Where do we get those cations and where do we get those anions from? They come from the fertilizer that you apply. For example, potassium nitrate. They are balanced charge-wise, so it's 1 potassium and 1 nitrate because it's a K plus and a nitrate minus. And you have 2 potassiums and 1 sulfate because a sulfate has 2 negative charges, so you need 2 potassiums to balance that. So you have the fertilizers, they are electrochemically balanced, and you apply them to get the correct balance of the ideal nutrient solution. You cannot just apply potassium on its own. It always comes with a counter ion. So you need to decide which is the best counter ion, because if I apply potassium, I need to make sure that I'm not upsetting the anions on the other side. Where do we get the fertilizer from? It's the soil solution. And where does the soil solution get the nutrients from? It's from the cation exchange capacity of the soil. So your soil has a certain amount of nutrients in it, built into it. You determine what they are from a soil analysis, and they become available and they feed the nutrient solution, the soil solution around the roots. And then also if there's any deficiency or any lack, then we apply fertilizer. You can see on the left-hand triangle, we have potassium, calcium, and magnesium, as I've said. But if we introduce excess amounts of ammonium, which is also a cation, or sodium, which is also a cation, it will disturb that balance. And that's the concept of useless salts. And they compete with the cations that are really important for plant growth. If we look at the anion side, the chloride, if you apply too much chloride, then you will also disturb the anion balance of the nitrate, the phosphate, and the sulfate. So that also can be depicted as a useless salt. Chloride is required in small amounts, I don't deny that, but when you apply excess amounts, it will certainly affect the anion balance. Another example of a useless salt is excess sulfate. Often one would say, I need a chloride-free product. So you apply all your potassium, which is required in the highest amount. All your potassium is potassium sulfate, but your plant requires very little sulfur. So the excess sulfate becomes a useless salt or a competing anion. Also in the plant nutrition, we will touch on it later, is foliar nutrition. I've just put it here because ideally, if you have an ideal nutrient solution around your roots, you shouldn't have any foliar application necessary. But during peak demand periods and high yields, you might want to add some foliars to that. Right, if we look at the different sources of potassium and how it affects this cation and anion balance. And we have potassium nitrate, we have potassium sulfate, potassium thiosulfate, and potassium chloride. And you can see that the potassium is the common denominator in all those, in those four products, but the counter ion is what differs. So potassium nitrate, you have a nitrogen or nitrate counter ion. In potassium sulfate, you have a sulfate and potassium thiosulfate, a thiosulfate counter ion, and in potassium chloride you have a chloride counter ion. Here's a picture showing chloride toxicity of various crops. There's a mandarin of interest to you and a citrus just showing chloride toxicity in the leaves. But besides direct chloride toxicity, chloride, because it's utilized in very, very small amounts, if you're over applying chloride, for example, as in potassium chloride, then you will affect the salinity around the root zone, which will affect water and nutrient uptake. But more than that, even if you don't have a chloride toxicity, you are definitely going to disturb the ideal nutrient solution balance. And again, when you introduce excess chloride, you influence the balance of the anions, the nitrate, the phosphate, and the sulfate with the excess chloride. If we look at potassium sulfate, the second potassium source, and we look at the extraction by an orange fruit, you can see that it requires 7 pounds of K2O per metric ton and only 0.3 of sulfur per metric ton. And that ratio of K2O to S is 1 to 0.04. If you fertilize that crop using potassium sulfate, you are introducing 8 times more sulfur than the plant requires. If you use potassium thiosulfate, you are introducing 17 times more sulfate than what the plant requires. That excess of sulfate definitely affects the soil salinity because it's in the soil solution, and it can produce or react with calcium to form gypsum and become less available. And it can also, or most definitely, will disturb the ideal nutrient solution or the anion balance. This graph shows the EC or relative EC on the y-axis, and it shows potassium nitrate as a potassium source. And to get this exactly the same potassium and sulfate, we use potassium sulfate and ammonium nitrate. And also to get the exact same potassium and nitrogen, we use potassium sulfate and ammonium sulfate. And you can see clearly that using the potassium sulfate and ammonium nitrate, you're almost 1.5 times higher EC in the soil solution. And if you use potassium sulfate and ammonium sulfate, you are almost double the EC in the nutrient solution. So that is with the same potassium and the same nitrogen. This is a graph showing an ideal nutrient solution in hydroponics in the Netherlands, and this is what they use for tomatoes and sweet peppers and cucumbers and melons. And the ideal nutrient solution here is nitrate. This is in parts per million or milligrams per liter. That the nitrogen is 224 parts per million, ammonium is 16 parts per million, potassium is 314 parts per million, and sulfur is 71 parts per million. That's what the grower is trying to achieve. Now, if you apply potassium nitrate, you can easily get this to get that correct balance of the nitrate and the potassium. And you will see that the nitrate to ammonium ratio is 93% nitrate and 7% ammonium. This is ideal for a hydroponic situation. In the soils, it's slightly more forgiving. You would go for perhaps 75% nitrate and 25% ammonium in the soil solution. Ammonium is not necessary as a plant nutrient. It is used in hydroponics purely as a pH regulator. If you apply potassium sulfate as your potassium source, it disturbs the balance because you have to apply it with a nitrogen form. So your sulfate is excessive. It goes from 71 to 161, which is now more than what the grower needs. So that's an excess sulfur, and it creates a saline solution. And your nitrate is under-applied because you're not using now potassium nitrate, and you have to apply a different N source, nitrogen source, which will then push up the ammonium, and the nitrate will be lacking. And your nitrate to ammonium ratio there is 58% nitrate and 42% ammonium. So you've got an excess of ammonium, and as I said with those triangles, as soon as you've got too much ammonium, you start affecting the cation balance of the calcium and the potassium and the magnesium. If we look at the different potassium sources again, we need to study the different effects of nitrate and ammonium on the plant. Remember, if you apply potassium nitrate, you're applying both potassium and nitrogen. If you apply potassium sulfate or potassium chloride, you need to add a nitrogen source. And if you add a nitrogen source, you have to use a urea or urea ammonium nitrate or ammonium nitrate and ammonium sulfate. The common denominator of all of those is that they contain or break down to ammonium. There might be some nitrate in those products, but you are introducing a whole lot more ammonium that you are not doing if you're using potassium nitrate. If we look firstly at urea application, if we look at the graph on the left, you have urea volatilization or losses to the atmosphere, and that is the y-axis is a percentage loss, and if the x-axis, it was tested over 3 different years, and you can see that by applying urea, which is the dark green bar, that you are getting losses to the atmosphere. If you look at the graph on the right, that amount or percentage of loss is definitely affected by soil temperature. So the percentage of nitrogen lost on the y-axis versus days on the x-axis, and you can see the 3 different temperatures shown that your greatest loss is with higher soil temperatures. So there is a real danger of losing nitrogen by volatilization, that's ammonia, to the air if you use urea applications. The reason is that urea converts to ammonium in the soil, but the first intermediary step is to form ammonia gas, and only under ideal conditions will the NH3 convert to NH4, which is ammonium, and there is a loss factor in that ammonia gas to the atmosphere. So the golden rule is try to avoid using urea and urea-containing fertilizer products, especially in high-value horticultural crops. You don't need to use urea at all. The other factor affected by using either nitrate or ammonium is the pH of the rhizosphere. Now, to understand the pH of the rhizosphere, or to define it, it is that portion of the soil that is directly affected by the roots. So you can have the bulk soil pH, and then you can have a pH different to the bulk soil pH that's been created by the influence of the root. So that zone of influence around a root is called the rhizosphere. If you strictly use ammonium fertilizer, you will decrease and acidify that rhizosphere pH dramatically. If you use nitrate fertilizer, you will increase the pH of that rhizosphere compared to the bulk soil pH. This picture I show you now, the left-hand portion are different colors depicting different pHs, so from yellow-orange to very, very dark red, a pH range of 4.5 to 6.5. Then the middle picture is the same plant, it just has a glass separation, where the left-hand side was used using nitrate fertilizer only, and you can see how the pH around those roots is sort of around 5.5 to 6, and the glass portion of that same plant root on the right-hand side was ammonia fed, and you can see how the pH has dropped to even less than 4.5. The guy who did the study is a guy called Romhelt, and he says that the difference between ammonium and nitrate-fed plants can be as much as 1.5 pH units around the roots or in the rhizosphere. And 1.5 pH units is a different, because of a log scale, between 10 and 100 times difference or more acidic around the roots using ammonia, which can lead to toxicities. The difference also between ammonium and nitrate nutrition will influence salinity stress in the soil. And if we look at this pictogram, there is a common antagonism between chloride and nitrate. So nitrate will be taken up into the plant and excess chloride will stay in the soil, which will definitely affect the salinity of the soil. There's also an synergy between ammonium and chloride, because the ammonium is a positive and the chloride is a negative. But we've seen that neither of those are essential in great amounts for plant nutrition. And so it creates all sorts of issues applying ammonium and applying chloride, because you will get a synergistic uptake of two elements that actually aren't that great for the plant. If we look at the synergistic and antagonistic relationships in slightly more detail, you'll see the picture on the left where the ammonium will compete with the other cations. As I said, from the ideal nutrient solution, your ammonium will compete with the uptake of calcium, magnesium and potassium. And if you look at the picture on the right, if you apply nitrate nitrogen, there's a synergistic uptake between your calcium, magnesium and your potassium. So the nitrate definitely enhances the uptake of the other cations into the plant. This is a graph from the International Potassium Institute, and it shows a barley plant, which is obviously not a citrus plant, but the concept is exactly the same, and it's the same for all crops. And you can see that the different potassium sources, potassium sulfate, potassium nitrate and potassium chloride, and you can see the synergy in the center two graphs are showing the synergy between the potassium and the nitrate uptake, and that's because they're both required in large amounts and they help each other into the plant. So it's a very good demonstration how the nitrate and other cations, in this case potassium, work together. This graph also shows the difference between nitrate and ammonium, but it's again just a model. It's using watermelon as the model, and the x-axis on the bottom is what's important. The left-hand part is 3:1 nitrate to ammonium. The middle portion is 1:1. That's equal ammonium and equal nitrate. And on the right-hand side is higher ammonium and lower nitrate of 1:3 nitrate to ammonium. And what you can see for the potassium and the calcium and the magnesium is the synergistic effect of the higher nitrate versus ammonium on the left. And as your ammonium increases, you can see that the uptake of your cations, potassium, calcium and magnesium, decreases dramatically. The differences in ammonium and nitrate nutrition will also affect the vulnerability of roots to heat stresses. This is a strawberry plant, and you can see the 4 pictures on the left were the plants that were fed with only ammonium nitrogen, and the 4 pictures on the right show the plants that were fed with only nitrate nitrogen. And the temperatures, the 4 pictures on the left show the temperatures ranging from 10 to 32 degrees, and similarly on the right. And what you will notice immediately with the ammonium fed plants at 32 degrees soil temperature, the roots and the plant are completely dead. Whereas using nitrate nutrition, you can see that the plant is still thriving. And the reason for that is that ammonium is metabolized to organic compounds in the roots exclusively. And one of the intermediary steps again is ammonia gas. And the second step of ammonia to organic compounds is blocked under high temperatures, and that ammonia gas in the roots is toxic to it, kills the roots and will kill the plant. You need to be aware of this if you have sections of orchard with certain soils that are very hot, and if you have roots fairly close to the surface, and if you feed ammonium, you're going to be sure to kill those roots in that higher temperature soil close to the surface. A very important aspect of the differences between ammonium and nitrate nitrogen sources is the energy efficiencies and carbohydrates within a plant. This picture shows very basically the nitrates are converted to organic compounds in the roots, but also in the leaves, primarily in the leaves. And that conversion, even though it goes via ammonium to organic compounds, is driven by light energy, and it is very efficient. Whereas ammonium, as I said previously, is converted to organic compounds only and exclusively in the roots. And it has to get its energy from carbohydrates, and those carbohydrates are carbohydrates coming down from the leaves, and so it is energy inefficient. Those carbohydrates could be going to growth and to fruit development or to storage organs, etc., etc. But it is here converting ammonium to organic compounds and being less efficient for the plant. This is a part I'll just read for you. Reduction of nitrate to nitrite and ammonium is predominantly taking place in the leaves of many plants. The critical step, reduction of nitrate, is driven by the energy of 6 electrons excited by the capturing of light in the chloroplast. The ammonium from this process is then incorporated into organic compounds, and this process is also using energy derived from the photosynthesis 2 in the chloroplast. This is energy efficient because it does not rely on the consumption of carbohydrates by the plant. Ammonia taken up by the roots is exclusively metabolized to organic compounds in the roots. This is achieved using NADH and NADPH derived from carbohydrate energy from the leaves via the phloem. This process always requires the consumption of carbohydrates and competes with other energy requiring processes such as root growth, cell survival, and there is a restriction in the availability of carbohydrates. This is energy inefficient and always leads to lower dry matter accumulation, affecting both yield and quality. Just to show you the effect of various K sources on the dry matter yield of tomatoes, tomatoes being the model crop here, we have on the y-axis metric tons per hectare, and on the x-axis we have a control. And then we have 3 different sources of potassium: potassium chloride, potassium sulfate, and potassium nitrate. And then we have on the right-hand bar just an increase in the potassium nitrate. But even in the 92 kilograms per hectare of K2O, you can see a slight increase in dry matter accumulation, in yield, metric tons per hectare, and also by increasing the potassium in the form of potassium nitrate, a dramatic increase in metric tons per hectare. The last factor or comparison between nitrate and ammonium is very important in saline growing conditions, and this was a trial done by Dr. Oosthuizen and co-author in South Africa, and it shows citrus seedlings that were grown in sand pots, and to that sand was added sodium chloride to make it more saline, and to other pots was added sodium chloride plus calcium carbonate to make it saline plus a higher pH. So they're the 2 treatments: the river sand plus sodium chloride factor 1, and also the river sand and sodium chloride and calcium carbonate. And then the second factor, we're using potassium chloride, potassium sulfate, and potassium nitrate as a potassium source. And then obviously the potassium chloride and the potassium sulfate had to have a different nitrogen source to make up for the nitrogen. So the K and N were exactly the same. And as we would expect, the potassium nitrate outperforms the potassium sulfate and the potassium chloride in both the saline sand and in the saline sand with a higher pH. And it all boils down to the ideal balanced, or the ideal nutrient solution, ideal balances around the roots, or even if you have a hydroponic situation or an orchard situation, you need to have that concept of balanced nutrition around the roots, the balances between the cations, the potassium, calcium, magnesium, and the balance between the anions of nitrate, phosphate, and sulfate. And if you keep that in optimal conditions, you are going to have a better yield and better dry matter production. If we briefly touch on the benefits of potassium as a foliar treatment, we say that it's not always required, but it might be required in certain situations. So if you look at this picture, the top left quadrant says that in periods of high nutrient demand, so that's after flowering and you have rapid cell expansion and cell growth, that's a period of high nutrient demand. and the roots might have decreased in growth and decreased in activity. So you would consider to spray foliar nutrition on at that stage. If you look at the top right-hand quadrant, if you've somehow missed your early fertilizer applications and you may be concerned about that, or if you've had excess rain or too much irrigation and nutrients are leached out and you've missed the peak to reapply, then you would consider to apply nutrients via the leaves. If you look at the bottom left quadrant, and there are certain nutrient imbalances in your soil, such as very high calcium and magnesium versus potassium, you would consider then to rectify that with potassium applications through the leaf. And the bottom right-hand quadrant, if you have other soil problems such as a non-ideal pH or very cold soils or low reserves or certain nutrient fixing problems, or too wet, then you would also require a foliar nutrition or consider a foliar nutrition. So we only use them when there is a recognized need, as discussed. Just to touch on a few applications of potassium nitrate. These first two graphs were, I'll show you the graph on the left. The yield in kilograms is on the y-axis, and various crops around the world on the x-axis, and the foliar potassium are the dark bars and the control are the light bars. And in all cases, and in the different crops shown, the orange in Israel, in South Africa, and the clementines in Spain, and the mandarins in Egypt, and in Chile, the oranges, you can see that where foliar potassium was applied, that there was an increase in yield in kilograms per tree. The graph on the right shows one application of potassium nitrate in marsh grapefruit. This was done in Florida, and you can see that there was an increase in fruit size, that's the y-axis, over time because of that foliar application of potassium nitrate. This author and this publication showed the differences in foliar leaf levels, and they showed a range from foliar potassium at 1% up to foliar potassium of 2%, and they noticed that there was a definite increase in fruit size as the potassium of the leaf increased. This was work done by University of California, Riverside, and it was foliar applications with potassium in orange, where they used potassium sulfate as a foliar and potassium nitrate compared to no potassium. And you can see that the boxes per tree increased with both foliar applications, the fruits per tree increased with both foliar applications, and you can also see that the fruit size increased, especially with potassium nitrate. So you definitely had a yield increase with both sources of potassium as a foliar, and an increase in fruit size with a potassium nitrate application. This work done by these authors with the effect of potassium nitrate sprays on Mineola, on the acid and sugar contents and fruit size of the Mineolas. And the foliar sprays with potassium nitrate significantly increased the acid and sugar contents of the citrus fruit. Moreover, the fruit size was significantly increased as well. So if you look at the table, you can see that the top line is the control, and the bottom line is 4 sprays of potassium nitrate, and the acid content increased with the potassium nitrate, and the sugar increased with potassium nitrate sprays, and the fruit diameter also increased with the potassium nitrate sprays. Now, if you calculate the sugar to acid ratio, it actually stayed the same at about 14, but the sugar in the control was so low that it would have affected the taste. It would have been insipid or pretty tasteless. So the foliar potassium actually increase the sugars to above the minimum level without affecting the sugar to acid ratio. This is work done by Bondman, 1995, where citrus fruit size can be increased with foliar sprays of potassium nitrate of concentrations of 3 to 6%. And it was done on Shamuti oranges, on ruby grapefruit, and on Nova tangerines. And if we look at the oranges first, the kilograms per tree yield increased with potassium nitrate. The juice content increased with potassium nitrate. And you can go through it and you can see that of the other two fruits as well, that the benefits of the potassium nitrate sprays were apparent. This is other work done by Bondman in 2001, also on oranges, Valencia oranges. And he sprayed 3 applications at 1% of potassium nitrate. And the number of fruits per tree increased by 29%. The TSS, or the total soluble solids, increased by 24%. The yield increased in metric ton per hectare by 28%. And most importantly, the gross income in dollars per hectare increased by 29%. A disease prevalent in the world and worse in some areas than others is the HLB virus. And it has been shown that sprays with potassium nitrate or potassium nitrate cocktails decrease the symptoms of the HLB virus. And that's by regenerating phloem cells and allowing photosynthesate transport. And you can see these two pictures of an HBL affected fruit and a normal shaped Valencia fruit. And the famous one is the Maury Boyd cocktail containing potassium nitrate. In these 3 pictures, it was a study and published in one of the Florida journals. And it was a 5-year study on the HLB virus and potassium nitrate sprays. And there were 3 potassium nitrate sprays at, in this case, 22.5 pounds per acre per spray in March, June, and September. And you can see from the pictures how the trees improved from 2008 to 2014. Dramatic improvement. So what are the general potassium nitrate recommendations if you've decided that you need to spray them? There are 5 moments of spraying. The first 2 are in the pre-bloom and post-bloom, so that's the fruit set and fruit retention stage. And the other 3 are in the fruit growth stage until color change, which will affect fruit size and fruit quality. And the recommended rate you can see in pounds per acre or in kilograms per hectare. And you will definitely get higher fruit set and increased fruit size, increasing your yield, and also better plant performances against specifically HLB. Just a few conclusions from today's presentation. We need to think which nutrients do you need to apply and how you're going to do that. You're going to look at your leaf analyses, you're going to look at your soil analyses, and you're going to decide what is the ideal nutrient solution around my roots, and you are going to apply the correct nutrients accordingly. You also need to think of how much to apply, and that will tell you, or you need to work that out again from your soil analyses, your leaf analyses, and the target yields that you are trying to achieve for your crop. You then need to think where to put the fertilizers, and you need to identify the area of active root growth and place it there. You need to think, when am I going to apply it, which phenological stage, and as we showed in some of the graphs, it's much better to put your fertilizer on earlier, because on a monthly basis your fertilizer is taken up definitely in the first half of the citrus growth stages, but to get it on even before that, so you don't miss that period of active uptake. And then we spent a lot of time on the form of nutrients, and we had the two triangles of the ideal nutrient solution and the balances of the cations and the anions. So you need to consider firstly that potassium is taken up in the greatest amounts, and then nitrogen. So it's best to put on your nitrogen as a nitrate because it will facilitate your potassium uptake, and also your calcium, which is your third most important element. So think ideal nutrient balance. And then lastly, to consider whether you need to apply foliar applications or not. Think of peak demand times. Do you have soil issues? Do you have pH problems? Is it too cold? Do you have imbalances? Or did you miss crucial fertilizer application times? Then you would consider to put on a foliar application. Thank you.